Hello, everyone, and welcome to our online lecture series for genetics. Today, we're leaving behind our discussions of the structure of genes, processes of forward and reverse genetics, and taking our Punnett squares to the practical. Rather than Punnett squares where they're theoretical, we're going to be looking at real histories in the form of pedigrees. Now, pedigrees, you may have heard this, it's a brand of dog food. It's a way of indicating parentage. It's essentially a chart that shows ancestry of a phenotype. In particular, pedigrees are looking at diseases. They're very useful when we lack genetic sequencing to identify traits within an individual. Fortunately, nowadays we're entering the era where sequencing can inform us a lot. But in absence of that, pedigree information is still fairly important, and there are lots of jobs out there as genetic counselors. So mastery of this material here and being able to digest what you see here, if you find this interesting, I would strongly encourage you to pursue a career as a genetic counselor, see what requirements are there. Ultimately, the goal of a pedigree for us is going to be to determine a mode of inheritance. How are these traits inherited in terms of, are they X-linked, are they autosomal, are they dominant, are they recessive, and what other forms of inheritance do they have? Now, you might think that this is all solved in terms of the human genome, but I can assure you that this is far from it. Every new allele can have a new mode of inheritance. Every new disease that we identify can have a new mode of inheritance. And there are lots of organisms that we have studied far less than humans. For example, my wife was working on corn plants, which we've studied extensively, but there are some traits whose pedigree we're still trying to figure out, and whose modes of inheritance we're trying to figure out, and whose alleles we're trying to figure out, and whose genes we're trying to figure out. So, that said, we first need to go over what the symbols are. So this is an image taken from your textbook. We see that males are indicated with a square, females with a circle. So males are square, females circle. Keep that in mind forever. An affected individual is going to be colored in black fully. Heterozygotes are carriers. Known heterozygotes, you won't always know this case, are going to be halfway colored in. And then a carrier of a sex-linked recessive trait may be colored in with a black dot. A dead individual is going to be crossed through. And then abortion or stillbirth is going to be indicated by a truncated or shorter line here. We see mating pairs with lines drawn between them. Parents are indicated by children with brackets, and they have an order of birth. We can indicate non-identical twins by one line creating two individuals, or identical twins as a triangle. If we don't know the sex, a diamond is shown. The individual being studied, or the propositus, is indicated with an arrow. And then ultimately we want to be able to determine the genotypes of individuals with these pedigrees. And then hopefully nothing we see in our pedigrees are these consanguineous marriages, meaning mating between siblings. They're indicated with double lines. Though you will see consanguineous marriages in the pedigrees of some of our ancient European royalty. It's not an uncommon thing. So keep in mind that as we work our way through this lecture, going through our examples of pedigrees, because really the way to do this is to look at examples. You should be pausing the lecture when shown a pedigree. You should try to determine what is the mode of inheritance for that trait. Once you have a hypothesis, then continue on. And as you go through solving all of these problems, our lectures are going to be more and more like problem sets as we go here. You should keep in mind two questions. Why does inbreeding matter in a pedigree, or for any trait of a disease? Why do we go yuck when we see these consanguineous marriages? And then, broader than that, why does ethnicity matter when talking about diseases? Why do we say things like sickle cell anemia occurs more commonly in African Americans than in Caucasian Americans? What kind of why does that occur, and why are pedigrees kind of the answer to that question? So I want to start us off by looking at this pedigree here, where we see a female. Remember, females are circular, mating with an affected male. We look at their children. 3 children produced: female unaffected, male affected, male affected. This one has no progeny for us to infer anything, but this affected male does. We see him mating with an unaffected female, and again, his males are affected, whereas the females are not. In the case of the unaffected female, that's a progeny of our affected male, you may expect that some of her children may be affected if she is indeed a carrier. We don't know that information, it is not specified, but we see that they're not. Therefore, what type of inheritance do you think is shown in this pedigree? Again, just pause that video for a sec. If you came to the conclusion that this is Y-linked, you are correct. Or at least, that's what I think too. It has all kinds of males being affected while females are not. And since the female doesn't pass it down to any of her progeny, including her male progeny, it means that this homogametic or XX female is not passing it down even though her father had it. Her father gave her the X that did not carry the trait. So this is an example of one of these pedigrees. And before diving into all the different types of pedigrees, I think we should first dive into the different types of inheritance and some of the diseases that we can expect to see with these different types of inheritance. So first and foremost are our autosomal dominant traits. Now, when considering our autosomal dominant traits, you're not going to see a difference in gender. So you're going to see both males and females impacted equally. You're going to see affected parents passing it on more likely to their children, meaning that if you have an affected parent, at least 50% of the children will be affected by the disease. Some of the examples of these autosomal dominant diseases, as well as some of the genetics behind it, because I think it's important that we tie the pedigrees to what we talked about before. Remember, we're really trying to understand the basis of what these mutations are doing on a molecular level and how we've studied them, how we've came to these conclusions. So first up, one that we don't have a picture for is pseudoachondroplasia. This is a disease that is a type of dwarfism. So it is a disease that impacts the development of limbs. It does not manifest up until something like 2 or 3 years of age because growth appears normal at first. But then there is a slowdown in linear growth compared to peers, so the individual will not get as tall or long as their peers, and then they may appear to waddle because of lower limb deformities. Now, it is a trait that is homozygous lethal, so you're not going to be able to have an individual that is homozygous dominant for this trait because they will not be able to make it to maturity. We do know a lot about pseudoachondroplasia. Its genetics is quite fascinating. It has been mapped to chromosome 19 in humans. Remember, we have 23 chromosomes. The particular gene that it's on is called the cartilage oligomeric matrix protein, or COMP protein, made of 19 exons, so kind of a big thing, with 757 amino acids. But as testament to the idea that you can get this disease with a lot of different mutations, therefore a lot of different alleles, we've found thus far something like 60 different alleles in this single gene that will cause pseudoachondroplasia. Kind of wild, right? Next up, we've got polydactyly, or the presence of supernumerary fingers and toes, meaning 6 fingers, 6 toes, extra fingers or toes. A trait that is controlled by many different genes and many different alleles, so nothing that we can point to directly, because these developmental type of traits have so many possible different causes. Yeah. The next one that I have a strong affinity for, partially because I think it is quite beautiful, and partially because I know a lot of organisms with this trait, is piebaldism. It is a disease that results in the lack of mature melanin-forming cells, meaning that the cells that cause pigmentation in these organisms don't form where they're supposed to. And it's oftentimes associated with two particular genes, KIT and SNAI2, SNAI2. Now, I point to SNAI2 because it's highly likely that at some point in this class I will go off on the tangent of piebaldism in dogs in particular, because we've studied it a lot and we understand that it has to do with gastrulation, or the proper formation of the gastrula. When you're a small ball of cells, the proper pinching in of your internal digestive system and the location of those cells. So it's a really fascinating thing, and it's named for the fact that the magpie has a bald spot or a white spot on it, and we see it in all kinds of different organisms. Keep in mind, birds and horses are quite distantly genetically related as far as vertebrates go, but they still have a lot of the same gastrulation genes, developmental genes, and melanin forming genes. Therefore, a mutation in these homologous genes in different organisms can result in the same phenotype, which is kind of neat, that the reason this bird and this horse are colored similarly is because they have the same gene that mutates in the same way, causing similar phenotypic shifts. Now, next up, we've got autosomal recessive diseases, the best known of which is sickle cell anemia, though it is far from the only. Now, autosomal recessive traits, in contrast to our autosomal dominant traits, will be much rarer in the pedigree. So you're going to see less affected individuals. But just like our autosomal dominant traits, you're also going to have no difference in genders. That's very important to keep in mind as we work our way through our practice problems. So sickle cell anemia is the result of a mutation in the hemoglobin gene, which, if you remember from Bio 1, you've got a couple of different hemoglobin subunits, so it's a mutation in a particular subunit. Now, interestingly, and this I find totally freaking fascinating, it seems to have arisen many different times. However, even though it's arisen many times, it seems to be, quote unquote, I'm making air quotes with my hands here, can't see, it seems to always be the same allele. That allele results in a GAG converting to a GTG, change in an amino acid in hemoglobin subunit A, to form hemoglobin subunit S, or sickle cell anemia here. What that subunit S does, differently from subunit A, is that under low levels of oxygen, it will form a fiber and begin to precipitate out. So you can actually see some of those sickle-celled red blood cells that are now, rather than being solubilized in hemoglobin that's still performing its job of grabbing oxygen, they will change their shape conformation. There's a lot more to be said about the prevalence of this disease in populations and potential adaptive traits, the overlap with malaria, because it's thought to be, well, there's good evidence to show that sickle cell disease corresponds to high rates of malaria, so it allows humans to have some adaptive advantage to malaria. No disease in human history has killed more people than malaria, which is kind of wild to consider. So it is also kind of not surprising to think that we might be arriving to a very similar mutation in many different areas if malaria is such a strong selective pressure. This would be a huge advantageous allele in those areas, but then not advantageous in areas where malaria is no longer present. For example, malaria used to be prevalent in southern Europe, particularly like Italy and um, Greece, and Spain as well, the Mediterranean regions, but it's no longer endemic there today. Its disappearance is within recent history. The Romans certainly dealt with a lot of malaria. So sickle cell anemia is a fascinating thing. Now, getting away from our autosomal traits, we have our X-linked traits. Some examples of X-linked dominant traits are X-linked hypophosphatemia, which is a disease that may be better known as vitamin D resistant rickets, which results in soft bone development in young children. So that's not good for their overall life. It results in the mutation of the PHEX gene, which governs fibroblast growth, which of course is present on the X chromosome. Now, when we look at other genes or other diseases that are X-linked in terms of pedigrees, we see that mothers are able to pass it down to both children, meaning both girls and boys. I'll actually just get rid of kids here and use the gender symbols here. Ooh. No, you're not going to let me? There we go. Whereas fathers that are affected are going to be passing it down preferentially to their girls, because father here is the gender determining gamete. The sperm determines the gender of the child. And if the father sends along a Y chromosome, it's not going to be carrying the disease on his X. Now, in X-linked recessive traits, you're going to see females very oftentimes be carriers of the disease, and the disease will manifest in boys. They will be affected. There are a lot more noted diseases in this category, things like red-green color blindness, which is, again, going to be accomplished with a lot of different potential mutations, simply because a lot of the genes for color vision are on that X chromosome. So you can mess them up in many different ways and still result in color blindness, as well as hemophilia, which is a very well-studied disease that has genetic causes in terms of mutations that will affect clotting factors. In fact, one of my second cousins, who lives in rural Poland, has hemophilia. But thankfully, these days we have not only good screening methods for it, but we have reasonable treatments for it. So this individual has no loss in quality of life as far as I've been able to hear. Now, for our last two modes of inheritance, we have some of the non-traditional ones. So you can imagine, hey, You can be dominant or recessive and found on autosomes, or you can be dominant and recessive and found on the X chromosome, and that's going to lead to different pedigrees. But you can also be found on the Y chromosome. Now, in Y chromosome linked diseases, you'll see it only in males, which is one of the telltale signs for this pedigree above, that we only see it in males and never in children of females, that pedigree showed a Y-linked trait. Now, we actually don't have a ton of diseases associated with this simply because the Y chromosome has so few. However, digging in for the bottom of the barrel, we found a form of hypertension that seems to be Y-linked in rats. There are many ways to get hypertension, but at least one of the versions of it is associated with being male in rats, which kind of a bummer. Who knows what truisms or what truths will be revealed once we learn more about the human genome and the Y-linked diseases there. And then plastid or mitochondrial. I bring up plastid not because humans have them, but I may show you a tree that is of trees, a pedigree chart that is of plants, and plants inherit chloroplasts, and their inheritance is similar to mitochondria. Mitochondria and plastids are both inherited in the cytoplasm of the ovum or the egg. So whatever is the genotype of the mother is going to be passed on to the children, meaning that an affected mother will pass it on to all kids. So it looks very particular in terms of how this disease manifests. Now, there are a couple of different diseases here, but one of the ones that's most likely to be recognized is a form of diabetes and deafness. And we've, of course, mapped this one out very, very closely. It occurs as a result of a point mutation at the 3,243rd base of the mitochondrial genome in a region that is supposed to make the tRNA that codes for leucine in the mitochondria. And it results in these mitochondria not being able to process some of their proteins properly. Not great, and of course is going to have an association with largely what we would perceive as disconnected phenotypes, like inability to process sugar in the form of diabetes, process sugar properly, and deafness. So kind of interesting, but is going to yield very particular looking pedigrees. So again, here I'm not going to be overemphasizing the particulars of these diseases, but rather wanted to give you some examples of how these work in practice, how a disease that is autosomal dominant will affect phenotypes and what kind of things we see, and then how diseases that are X-linked, as well as Y-linked and plastid-linked, can manifest in terms of phenotype. But what we're going to do now is take some time to look over some pedigrees. So please, as we go forth here, pause the video with every pedigree and try to solve what form of these 6 different modes of inheritance are being shown in the next couple of pedigrees. Keep in mind, we've done Y-linked. So first up, let's take a look here. So take a moment and pause the video here and see if you can determine which mode of inheritance is shown in this pedigree, and then we'll take a stab through the logic ourselves. So hopefully you got a chance. Let's see what kind of things I notice when I'm looking at this. Well, first and foremost, does this differ in the genders? We have males carrying it, females carrying it, so we have equal ratios there. Do we know anything from whether an affected individual, do we see it in their children, or are any of these going to be carriers? Well, simply because of the fact that we know that none of the children have it from this affected father, we're assuming that this is going to be some sort of recessive disease, right? Same thing goes for the other side here. No affected parent, and yet 2 affected children. We assume that these 2 parents here, this next generation, the 3rd generation, we can start to deduce what their genotypes are. They result in children that are affected for a recessive disease, meaning that each of them would be a carrier. So let's go ahead and use letters here. I like big A, big A, little a. And therefore, you could do some math to try to figure out what is the likelihood that the unaffected child 1 versus child 2, with child 2 being affected, you know that its genotype is going to be little a. What is the likelihood that child 1 is a carrier? Well, for that, you have to revisit your idea of Punnett squares. So if we have big A, little a, crossing big A, little a, how many of these possible squares are going to be heterozygotes? Well, this one ends up being heterozygous, and this one ends up being heterozygous. But we know that this individual is not homozygous recessive. So what is the probability that individual 1 is going to be a carrier? It's not 1/2. It is instead 2/3, because we know that they are not homozygous recessive. And in this way, you're going to be combining some of the math that we remember from some of our Punnett squares into our pedigrees. A little bit of a taste of what's to come in some of the problem sets. So we've decided that this is an autosomal recessive pedigree. Next up, let's take a look at this and tell me what you think. So again, hopefully you've paused and got a chance to do some thinking on this. First thing I notice, we've got girls affected, and we've got some boys. We've got a situation where we don't see, we see that when mother has it, all of her children have it. When mother has it, all of her children have it. That right there, that statement in and of itself suggests to me that it is mitochondrial. Now, how would you write out some of the genotypes attached to mitochondrial traits or mitochondrial diseases? Well, this is another case where they're hemizygous. So I would maybe say mitochondrial, negative for having the affected trait, mitochondrial, positive, or wild type. All of her children end up being mitochondrial, negative. When mother is mitochondrial positive, she passes on mitochondrial positive or mitochondrial wild type to all of her children. So this is a good example of a tree for either plastid or mitochondrial inheritance, a maternal form of inheritance. Next up, take a moment. All right. So again, we see more ladies than men. We see when mom's got it, half of her children have it, and it's girls in this case. When mom's got it, a little over half of her children have it. In the pedigree where it doesn't exist, but there are no affected, we see nobody having it. It does not skip a generation. With all that information, which mode of inheritance would you guess it is? If you guessed autosomal dominant, then I'd agree with you. The fact that it does not skip and it does not exist, does not skip generations, and it does not exist in a tree that there are no affected individuals would suggest that it's dominant. The fact that women have it at a higher frequency in this pedigree than men would suggest that it is not X-linked, right? Because if it was X-linked, we would expect this male child from this affected mother to have it. The fact that that male does not means that it is not on the X chromosome. And we know for a fact that it's not on the Y chromosome because, hey, guess what? 4 ladies have it. So you have to both look at positive lines of evidence and negative lines of evidence in order to get a better picture of what mode of inheritance this disease is following. Next up, take a moment here and tell me what you think. So, we see 1, 2, 3 females, 1, 2, 3, 4, 5 males. So there's a little bit of a gender skew here. We see more males than females. We see when a mom has it, all of her boys have it, and none of her girls have it. In the situation where mom doesn't have it, but may be a carrier, we see that her boy has it, but her girl doesn't when her husband is unaffected. And then in the situation where both parents are affected, all children have it, whether they're boys or girls. This, to me, would suggest that the trait is X-linked. And then because it does not exist in the females, they instead are carriers, that this trait is X-linked recessive. All right, last one in this series. Take a look, tell me what you think. All right, so we see 1, 2, 3, 4 females, 1 male. We see a situation where 1/2 of the progeny are affected when one parent is affected. We see that when a father has it, he does not pass it on to his sons, but definitely his daughters have it. Remember, dad is giving the X chromosome. So he's passing on an affected X chromosome, it would seem, to these children, unless this is autosomal. Well, the fact that we're seeing it be present in one gender versus another when father has it is a pretty good tip that this should be some sort of X-linked trait. And the fact that mom doesn't have it, but her daughters do, would suggest that it is X-linked and dominant. And so these trees are excellent resources in terms of seeing some of the basic pedigrees, short, 3-generation pedigrees for our major modes of inheritance. X-linked dominant, X-linked recessive, both looking at gender skews, though we see with X-linked recessive that males are more likely to have it, whereas in X-linked dominant, we see that there's a correlation as to whether if your dad is affected, the sons won't get it, but the daughters will if it's dominant. And then if your mom has it, has equal chance of giving to children. We then have autosomal dominant, where they're about 50-50 ratios with no skipping. Mitochondrial inheritance, where mother is passing it along. And autosomal recessive, where it does seem to skip some generations. Guess I forgot to point that one out there. And then, of course, the one that we started with, are Y-linked recessive. Or Y-linked, neither recessive nor dominant, rather hemizygous. So now, with these under wraps, we're going to take a stab at some other practice problem sets. Though I will give this one aside. There are a series of phenomenal Wikipedia pages on X-linked recessive inheritance, X-linked dominant inheritance, dominance in genetics. It shows a wide array of both different traits. You'll see that I certainly pilfered some of Wikipedia's information to help supplement some of this lecture material in terms of the diseases in particular. But the Wikipedia pages surrounding modes of inheritance are actually fantastically helpful. So I would encourage you to look at those if you have some time. Let's get to some of our practice problems from the textbook. So first up, we have this giant pedigree. And the first question about this pedigree is determine the mode of inheritance. So again, take a nice longer pause and think of all the possible modes of inheritance given this pedigree. So, let's try to glean together the information that we have. Well, we see one side of the family where individuals are unaffected, have no individuals that are affected. When a male that is affected is incorporated into the family tree, we then get a whole bunch of individuals that are affected, right? This, to me, would suggest that the disease is dominant, right? In places where we see no affected individuals, we see no other affected individuals further along the family tree. But when one is introduced, it starts masking other traits. Very telltale that this is some sort of dominant phenotype. The other thing that you'll note is that when dad's got it, how many of his sons have it? Well, these two sons don't have it. Let's take another look at a case where dad's got it. Oh, I should change color here. Change it to red, sorry. So when dad's got it, son doesn't have it, son doesn't have it. When dad's got it, son doesn't have it, son doesn't have it. When dad's got it, son doesn't have it. Is it because dad's giving a Y chromosome to the sons, whereas the daughters receive his X? That's what I would bet. So the likely mode of inheritance is X-linked dominant. Now, there could be a couple others that would be possible, notably autosomal recessive, but it's not really possible with this tree, with this pedigree, rather. This would really cause, if we were to assume that this was autosomal recessive, there would be lots of, we see lots of gender bias. We see, data points that are not consistent with it being recessive rather than dominant. And so it's not really a likely outcome. And so I would only accept this as an X-linked dominant, or the most likely situation being X-linked dominant. But we can do a little bit more with this tree. I want y'all to try to build the Punnett squares for a cross of individual 1 and 9. So we're looking at the F4s, the fourth generation here. And we're going to be performing some cousin marriages. If individuals 1 and 9 mated, what would be the ratios of their offspring? If individuals 1 and 4 mated, what would be their ratios for their offspring? If individuals 2 and 3 and 2 and 8. You're going to have to use the information about the parents to help illuminate what's happening. Again, give the video a pause, and then join me when you're done with all 4 of these Punnett squares. Okay, so with the first one, we know that individual number 1 must be heterozygous because she obtained a regular allele from her mother and got the affected allele from her father. So let's go ahead and indicate how we're going to annotate our alleles. So X affected is going to be the affected individual or unaffected allele. And since if you have a capital A with an X, you carry the trait because it's X-linked dominant. And then X lowercase a is going to be wild type. Y is going to be our allele notation for having a Y chromosome. So individual 1, so let's take this here and draw the Punnett square for this. We're going to have mom on the top axis, dad on the side. Looking back at this for individual number 1, mom doesn't have it, dad does. So that means this XX lady is going to be affected from dad and wild type from mom. And then she is mating with individual number 9, who is known to be affected. So this individual is going to have X affected and Y. Now, let's say, regardless of gender, what is the ratio of progeny that she would expect to have affected by this disease? So let's go ahead and color these in here. So this is an affected female, this is an affected female, this is an affected male, and this is an unaffected male. So fully, 3/4 of her progeny would be affected. And hopefully you can see how I can twist some of these questions in order to determine how many of her males would be affected, how many of her sons would be affected, how many of her daughters would be affected, etc. But for now, we'll just go through the rest of these and draw out the Punnett squares. Next up, for 1 and 4, we already know that the female here is going to have our same X affected, X unaffected genotype, heterozygous genotype. What's individual number 4 look like? Well, number 4 is an unaffected male. So that means X unaffected, Y. Here we've got a heterozygous female that would be affected, a homozygous unaffected or wild type female, an affected male, and a wild type male. So here you can get both affected males and females and unaffected males and females from cross number 4. Next up, 2 and 3. Well, 2 is a male that is unaffected. So if we're going to draw the male here first, 2 is going to be a male that is unaffected, X lowercase a y, and 3 is a female that is affected, created from a cross of an affected male and an unaffected female, meaning that this individual is heterozygous. This Punnett square is going to look the same as our Punnett square for 1 and 4. Save the time filling that in. And I'll do our last question here. Individuals 2 and 8. An unaffected male crossing an unaffected female. Well, if no one's affected, and this is a dominant disease, well, then there will be no affected offspring. And you may get some of those questions. Hopefully, in doing some of these practice problems, I've illustrated to you what may be asked of you in terms of determining frequency ratios using the product rule in our Punnett squares. So a combination of, product rule in our pedigrees, a combination of Punnett squares and pedigrees at the same time. There are a couple other permutations of these problems. For the next two, the last two problems that we'll be doing in this lecture, we're going to be drawing again from the book for a couple of practice problems. The first problem that we're going to be doing states that there is an allele, lowercase c, that causes albinism in mice. The albinism means that they are white, whereas a capital letter C means that they are black. C for coat color in this case. We cross a big C, little c, against a little c, and we make 10 babies. My question of you is, what is the probability of all of them being black? Take a second and think about it. You should start with that Punnett square, right? So let's go ahead and draw a Punnett square. Big C, with little c. This individual is heterozygous. This individual is heterozygous. This individual is homozygous. This individual is homozygous. So with any given Punnett square, half of the progeny are black. And this question is asking, what's the probability that you make 10 children that are all black? In order to determine that, you have to use either the sum rule or the product rule. Now, for those of you who may not remember this from statistics or previous biology, the product rule, meaning that you multiply the probabilities, is used when there are independent events. The sum rule is used when there are mutually exclusive, meaning that having one black progeny would reduce the likelihood of having a second black progeny. Well, since that's not the case in genetics, most of the time we're going to be using the product rule here, we see that we're going to want 10 independent draws. Well, if we have a 1/2 chance of making a black mouse in one baby, what's the likelihood of having 10 of them? Well, 1/2 to the 10th, whatever small fraction that is. Hopefully that makes sense to you. And again, there are more practice problems in the book, as well as in our upcoming practice problem sets. So just be aware that this simple math is something that can be asked of you. The last problem that I'll ask you to take a look at today goes as follows. We're dealing with our hypophosphatemia. So let's go ahead and scroll on up. I remember hearing that word. Disease is hypophosphatemia. Oop, we got it as an X-linked dominant trait. So we've got with the disease of hypophosphatemia, we have a male that has it. Scroll back down some space here. So hypophosphatemia, male that is affected, crossing with a female that is wild type or does not carry it. What proportion of their sons will have hypophosphatemia? Well, we can go ahead and make a Punnett square for this, or a pedigree. Kind of the same thing. Well, if the male has it, put H for hypophosphatemia. Why? Should always indicate the nomenclature for your system. XH is affected. I write affected here because I'm too lazy to write out hypophosphatemia again. X plus equals wild type. And then Y is equal to male. Because we know that hypophosphatemia is a X-linked dominant trait. So the males got it. The female is wild type. The male will give hypophosphatemia to his daughters. And to his sons, he just gives a Y. This means, yes indeed, kind of a trick question. What proportion of their sons will have hypophosphatemia? The proportion is 0. If you're asking for what proportion of their progeny all in all, it is 1/2. If you are asking what proportion of their girls, it is 100%. So in this way, you're going to be combining a little bit of math, a little bit of genetics, and solving a lot of different pedigree problems. Hope you enjoyed our foray into pedigrees, and I do encourage you to look back at those Wikipedia pages. Disease inheritance is quite fascinating, and you should definitely fall down that rabbit hole of trying to see which genes play a role and if we know of any of the alleles that cause these different genes. It should hopefully expand your understanding to see that there are no simple ways to be purple or white flowers. Purple being wild type, maybe there's 30 different alleles for white flowered pea plants. I'll see y'all next time.